Alright, so 314 and 315 are probably one of the hardest concepts in this whole section, or this whole class to understand. So you need to make sure as you're going through that you ask questions and make sure you understand things before you just keep moving on. And also you have to make sure you don't get confused because there's many different parts of this, and because you're going to do first, you're going to look at what a microarray is, then you're going to predict what be shown in a microarray of a smoker versus a non-smoker, and then you're actually going to do a microarray. Now the tricky part when you do the microarray is that the colors don't actually match up from what you learned about. And so you just need to make sure you ask questions as you go through. So you need to know why we use what microarrays do and how they work, and then basically how to analyze the sample and view the results. So I'm going to go through the basics of what you need to know in order to figure this out. So DNA microarray, big long vocab term, but basically it's just the analysis of gene expression. So it's taking DNA samples and seeing how the gene is expressed. So what that means is the DNA on or off. So basically what we're trying to do with this is we figured out how we diagnose cancer. We look for tumors with radiology, and then if we find tumors we do a biopsy on them to see if they're actually cancerous or not by looking at the cells. And now what we can do is if people have cancer or if they are thinking they've developed cancer, we can do these microarrays to kind of figure out what genes are actually causing this cancer so we can better diagnose people and better treat people. But of course, you know, there are several genes that can cause cancer. It's a multifactorial disease, and so what we're gonna have. So this is still very complicated, and this might mean exact science because even this microarray assumes something, which we'll talk about in a second. But we're not going to focus on the environmental impact of it either. We're just looking at the pure genetic impact. Now the environment affects the genes and causes the genes to be maybe expressed, turned on and off, and that's called epigenetics, which we're not going to get into in this class. But we're mainly just going to focus on the genes and how you read whether or not they're on and off and what that means for cancer. Alright, so also when when we talk about this, you need to remember that tumor suppressors should be off or not expressed in a cancerous cell. Alright, so any genes that are turned off, they're probably tumor suppressors because we want them on in a normal cell. And then oncogenes are probably on in a cancer cell and off in a normal cell because oncogenes cause sporadic cell division. Alright, so you're also going to have to remember that as we go through this. So first, let's just talk about gene expression. So cells. Every cell in your body has the exact same DNA. They all came from one single cell from the fertilized egg, and then they all divided. But what happens as cells divide, they differentiate into various different cells, and this is only a small sampling here. And you see how they're all very different sizes, shapes, they all have different functions. That's all because of gene expression. So in blood cells, they're going to have the gene for hemoglobin on. All the rest of these cells have the gene for hemoglobin, but it's not going to be on. It's going to be turned off because they don't need hemoglobin. Only these guys do because they're the ones that carry oxygen around the body. Surface skin cells are going to have the gene for melanin on. The rest of them have the gene for melanin, but it's going to be turned off because none of them need melanin to protect them from the sun, and so on and so forth. You can go on and talk about all these genes. Now, most of these are all going to have some genes that are all turned on. So, for example, the gene for cell division, just so these cells can divide, will all be turned on. Muscle cells, these will all have the genes for actin and myosin because they all need actin and myosin to contract, if you remember from HBS. So all three of these would have very similar genes turned on, but neither of these would have actin and myosin turned on. So it's all about what genes are turned on and off, and that's what we're going to look at in cancer. So I basically just talked about this on the last slide. So, and that's why stem cells are so important. So if you've heard about stem cells are the ones that are basic unknown cell that then can differentiate to various other tissues by turning on and off those genes. So that's why stem cell research is so important, because if we can make these stem cells turn into these different tissues and grow tissues, we can then use that to treat people. Alright, so now let's talk about a little bit more basic information. So, if a gene is turned on, what that means is it's going to make a protein. Well, it's really hard to measure the amount of protein in somebody if you're looking at that, because first of all, proteins go outside the cell. So we want to look at something that's inside the cell. So what we do is we look at the intermediate step, and DNA's hard because it's so big. Alright, mRNA is perfect. It's already basically the copy of the gene that's being made, and then that goes outside to make the protein, but it stays inside the cell. So if we assume that if the gene is on, it's going to make mRNA and it's going to be present, then we'll assume every time you get an mRNA, the gene is on. If there is no mRNA, we're going to assume the gene is off, meaning the gene isn't even being transcribed into mRNA, so no protein is going to be made. Alright, so in skin cells, if we take that melanin, for example, in skin cells, you would see the mRNA that would be present to make the melanin, so these amino acids can be put together to make the melanin. But in nerve cells, there would be no mRNA. Alright, so this part wouldn't happen. The DNA would stay closed here. There would be no transcription of mRNA, so then there's no protein being made. Now we can take this a step further, which we're going to talk more about in 315, is that we can also measure the amount of mRNA that's being made. So we're going to assume that if you think about skin cells of darker people have more melanin. That's why they're darker. So we would assume that the mRNA, there's more mRNA being made so they can make more melanin. Whereas skin of light people would have just a few mRNA, so there's less melanin being produced. Alright, so now these are some assumptions. Now, some problems with this, and why it's not completely fail-safe, is that mRNA could be produced, but then maybe this doesn't happen. The mRNA doesn't actually go to the ribosome and get produced. So there might be mRNA in the cell, but it's still not making a protein. Alright, so there's various other factors that could happen that even though mRNA is there, maybe it's still not making the protein. So that's the kind of the downfall of microRNA, is that assumption that every time there's an mRNA, a protein gets made. Alright, so now, how do we measure how much mRNA is in our body? Well, we can actually do this really cool on a DNA microarray, and we can actually test for thousands of genes at one time. So this one only shows like 20, but this, as you can see here, throws like 1,000 genes on here. Alright, so what we do is we've already sequenced the whole genome. We know the DNA sequence for every single gene in our body. So what we do is we take that sequence and we make it and we put it on little wells, little tiny wells on each plate. So each one of these is a different gene. So I'm just, for the sake of argument, this is going to be gene 1, and maybe that's for melanin. Gene 2, maybe that's for actin. 3, maybe that's for hemoglobin. Gene 4. Alright, and then so on and so forth. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. Alright, so what these are all the DNA. So what happens is these are single-stranded here. And remember, mRNA is single-stranded. Alright, so if here's our little mRNA samples. So what can happen is if this is complementary DNA to the mRNA will stick to this as it flows over the well. And then if the mRNA is present, it will stick. And then if it's not, it doesn't stick. So for example, in gene 5 here, there's only the black piece. You notice how there's a black piece on every single one? This is the DNA for this gene. So let's say this is the gene for myosin. Alright, so we're going to assume that this is the gene for myosin. There is no mRNA attached to it. There's no mRNA for this cell or this cell, and I'll talk about what these mean in a second. So that means we're going to assume that neither of these cells produce myosin. All right, because there was nothing attached to this. There was no mRNA present, no mRNA to attach to this, so myosin is not made. So these are probably not muscle cells. Now, we have the control sample and the experimental sample. In cancer cells, this is usually the cancer cell, and this is your normal cell, because you want to compare the two. So what we're going to see is that in gene 4, whatever gene this might be, this one is made in the cancer cell. This one is off in the normal cell. So that's different between these two cells. So possibly this one could be something causing cancer. All right, because it's only made in cancer cells. So what is always turned on in cancer is this might be an oncogene here, because it's turned on here because it's only in the cancer cell. All right, so if this wasn't cancer and regular cells, let's just say this is a nerve cell and this is a skin cell, this would be probably for a gene that is only in a nerve cell. All right, like something to make the sodium pump. This one is only in the green cell, all right, because there's only the green attached to it. So what this means is that this is probably a gene that is only found in this one. So this could be, if we're talking about normal and cancer cells again, this is probably a tumor suppressor gene, because tumor suppressors are off in cancer cells but on in normal cells. So this is being produced, mRNA is being produced because it's on. This is our skin and nervous cell. This is probably melanin. Melanin's only going to be produced in our skin, but it's not produced in our nervous cells, so that doesn't attach to. So now let's look at this special one right here. All right, this one has green and red. This one and this one, these two. All right, these aren't necessarily special, but they're different than the rest of them, that they have two attached to them. That's probably because they both have that gene. All right, it's being made in this cell and this cell. So this is probably something that is not causing cancer if you're controlling normal and cancer cells, because they're found in both. So it's no different between either one. So they'll probably not have to do anything with cancer. And then if this is our skin versus nervous cell, that means it's something that both the skin and the nervous cell need. So something like a insulin receptor, maybe, because they both need to get glucose into the cell for energy. So maybe this is the gene for the insulin receptor. It could be just a gene for normal cell division. It could be a gene to make a ribosome. All right, so something that they would both need is there. So actually you would see a lot of these. All right, and then we can look at these thousands. So what you see here is little red dots. Those are the ones that are only found in this one. And then you see the little green dots. That one's found only in this one. And then you see yellow. That's what happens when they both mix. Red and green make yellow. And then black are all these, where the gene is not turned on in either cell. All right, so you have to remember those colors. And you'll go through this whole thing. All right, so that's pretty much a microarray. You just need to understand that there's already DNA for the genes on there, and what they do is that the mRNA hooks up to those DNA if the gene is turned on. All right, so what you're going to have to do is first describe how microarrays are performed. You're going to go through a simulation lab and be able to describe how they work, be able to interpret the results of an mRNA. So know what genes are on, off, not related, so meaning are not there, or they're expressed equally in cancer and non-cancerous cells. So you're going to have to know those colors that occur. And then you're going to, Grandpa Joe is Sue and Kevin's grandpa, and he's a smoker, and they're trying to convince him to quit. So what they're going to do is get a microarray done to see if he might have the genes for cancer.